Okay, next one. Okay, next one. 158 questions. During PCR, primers binds to the DNA strand in the which steps? So this questions which is framed from biotechnology. Technology. In biotechnology, framed from tools of RE-NEET technology. Tools of RE-NEET. So in which especially focus on PCR. So here we need to know some importance of PCRs. Here PCR means polymerase chain reaction. So this PCR technique developed by a scientist called Carey Mullis in 1983. And what is PCR? Generally PCR used to make millions and or billions of copies of specific DNA segment. So here PCR used to make millions or billions of copies of specific DNA pigment. So we use this PCR techniques, or simply we can know that. So it is used to amplification of our gene of interest. Amplification of gene of interest. We use this PCR techniques. Generally in PCR there are three important steps. There are three major steps are involved in PCRs. So in that first one, in denaturation, what happened here? So here at a high temperature, like at high temperature, approximately we use 94 degrees Celsius, and this high temperature will separate the double-stranded DNA templates. Separate the double-stranded DNA segment like this. So if you have 5' to 3' here, 3' to 5'. So these two strands are connected to each other with the help of hydrogen bond like this. Okay, like this. Hydrogen bond is present between these two strands. So at high temperature, 94 degrees Celsius, these two hydrogen bond break down and they become separated like this. They become separated. So 5' to 3' to 5. So the separation of DNA strands templates at high temperature, the process is called denaturation. Now after separation or denaturation, then it enter into next step, that is annealing steps. Annealing. So in annealing, what happened here? So annealing at lower temperature. So here the approximate around temperature is 55 degrees Celsius temperature around, in which what happened? This allows two sets of oligonucleotide primers. Very important. Here only oligonucleotide primers are added. Oligonucleotide primers are what happens? Bind to the complementary sequence of the single strand of DNA template. Bind to the complementary sequence on the bind to the complementary sequence on the single strand of DNA template. How means we will discuss single strand of DNA template. So here, for example, so there is a addition of here. So like that here at the each strands bind with the oligonucleotide primers here like this. So here and this side at the 3' end, the oligonucleotide primers are binded to their complementary single-stranded DNA. Like that, primer is added here. Primer is added here also. Primer is added. So this addition of or binding of primers to the 3' end of the each single-stranded of DNA, in which process? Annealing process. Okay. So then here next process. So and the third process is called extension process. So in extension, what happened here? Generally extension occur at around temperature 75 degrees temperature. Extension. So here what happened in around this at temperature, the tag polymerase, very important, tag polymerase enzyme play important role in this extension process. Here tag polymerase enzyme nothing but here thermus aquaticus enzymes. So the thermus aquaticus enzymes play important role in extension process. How means this tag polymerase enzyme which extend these primers here. So here this thermus aquaticus enzymes what will do? Which extend the primers. Which extend the what? Primers by using deoxyribonucleotide phosphate. So here we have various DNTPs like adenine, adenosine, guanine, cytosine, thymine. Like that, we have different DNTPs. So this DNTPs which is added by to the primers by tag polymerase enzyme. How means we will discuss here. So for example, take another. So here this with the help of tag polymerase here to the primer. For example, here added A, then G, then C, then T, like that. So here also added to the complementary. T means here A, C means G means C, A means T, like this. So addition of this DNTPs here, they are all DN. So addition of this DNTPs to the primer with the help of tag polymerase enzymes. So this process is known as extension. Like that, by the end of this complete extension of this, then finally they form from one double-stranded DNA. There is a formation of two daughter double-stranded DNA. By end of the here, like completely one strand will be like this. Two strands will be formed by the end of this extension process. Okay. So like that, each and every steps which is occurring PCR techniques. So here we need to identify so which statement is correct. So here in questions what they are asking, just wait. Yeah. So here during PCR primers here. Yeah. So here during PCR primers are binded to DNA strands in the which steps here, in which step the primers are bind to the DNA strand template, DNA template strand, in which step, annealing steps. Okay. So here the right answer is annealing. So here annealing is the right answer. First one is the right answer. Okay. So in here already we discussed. So in annealing steps only at lower temperature, and which allow the binding of oligonucleotide primer to the complementary sequence of DNA strands here only. So in annealing. So right answer is annealing. So next one, 159, match the following questions. So in list one, giving different features of this here with respect to their enzymes are different matter. So here under this question, transformation, cloning site, selection, ORI, all these are comes from biotechnology. In biotechnology, they mainly focus on tools of RDNA technology, in which these options are given from futures of plasmids or vectors. So from which these questions which frame. So let's start what exactly all these things. So here we need to remember the cloning vector, that is plasmid features. So cloning vector. Here cloning vector is what? Plasmid. So first we will discuss the future of cloning vector about plasmid. So generally plasmid has four important features. So in that first and most important, it has ORI site, ORI sequence. So here ORI means origin of replication sequence. Origin of replication. If you draw one plasmid like this, plasmid is an extra chromosomal single double-stranded DNA like that plasmid. Okay, so this is the structure of plasmid. So in this plasmid, there is a one important site is present here. So this site is called ORI site or ORI sequence, origin of replications. So here the importance of ORI cycles, the sequence which replication start from here only. Why? Because plasmids are self-replicating. Hence, the replication will start from this site only, ORI site only. So here I am writing. So ORI site means the sequence where replication will start. The sequence where replication will start, such site is called ORI site. Now one more, plasmid also has one more important features. So that is cloning sites. Cloning site, very important site here. This cloning site. So generally here plasmid has number of sites. So what exactly cloning site means? The site is present in various places. All these are cloning sites with different restriction enzymes. Okay. So here the the cloning sites are the specific recognition sequence, or also called specific recognition sequence, also called palindromic sequence. So where restriction enzymes which got the vector Y to insert our desired gene or gene of interest, or also we can use another term foreign gene or foreign DNA. So here, for example, we have various restriction enzymes which cut the plasmid here, and this restriction enzyme which cut only specific recognition sequence only. So if we cut here the specific recognition sequence by restriction endonuclease, so where we have some space, so in that space only our gene of interest get inserted. So that site is called cloning sites. And one more features of cloning vector here we have, so that is selectable marker, very important, select marker. So generally plasmid you has two important selectable marker here, like so for example this region, this side region. So these two are selectable marker region. So what exactly selectable marker means? Here the genes, selectable markers are nothing but made up of genes. So the gene such as antibiotic resistant genes. So which genes here? Antibiotic gene. So antibiotic resistant genes use, why you added this antibiotic resistant genes in this plasmid means, which is used to distinguish and selection of what? host from non-transformant, very important, from non-host. So here why we use selectable marker means, so we have to distinguish which is transformant host and which is non-transformant host, and also we need to select only transformant host. Why? Because what exactly mean of transparent host means, if the host which contain or DNA, recombinant DNA, such hosts are called transformant host. Here mostly host are bacteria. So most widely used bacteria here, Escherichia coli, E. coli bacteria we use as a host. If the host which contain rDNA, such hosts are called transformant host. The host which does not contain any rDNA, so in which no rDNA, such host is called non-transformant host. So how we identify which is, how we distinguish which is transformant, which is non-transformant means, because of having this selectable marker. So this selectable marker which distinguish which is transformant host, which is non-transformant host, and then after we need to select only transformant host. It means that this transformant host which contain our DNA genes. Okay, so for that purpose we use selectable markers. Okay, so like that we have various features of plasmids. So once we analyze this statement here, so transformation. So here transformation, first we read restriction enzymes. So who play important role in restriction enzyme means cloning sites. So here B is 1. So here ori transfer of DNA to host bacteria is called transformation process. Yes. So we have to transfer DNA or DNA to the host bacterium called transfer, means A2. So what is transformation? So transfer of DNA. Which DNA? Or DNA mostly. Okay, so transform of DNA into host bacterium, the process called transformation. So here we have to selection. So what we have to select here, so we need to select anti-selectable marker used as a antibiotic. So C is fourth one, and ORI sequence for replication. So like that, first two, here where we have two, C here we have two. Okay, so we need to remove all this, we need to eliminate. So 2 and B 1 and C 4 and D 3. So right answer is third one. So next one, 160, adaptive radiation. Here adaptive radiation in placental mammals and Australian marsupials, leading to similarity, yes, between distant species is an example of. Here founder effect, genetic drift, divergent evolution, convergent evolutions. So first, these questions which is framed from evolution. So in evolution, which is mainly focused in evolution. So this convergent evolution mainly led by the analogous organ. So just now we discussed in analogous organ, what is mean of analogous organ? These organs have same function but different origin. Same function and different origin, so that leads to formation of convergent evolutions. So here in which convergent evolution they are asking about adaptive, analogous, adaptive radiation. So what this adaptive radiation is saying, so this adaptive radiation, it states when more than one, here it states that when more than one adaptive radiation occur in isolated geographical area. Isolated geographical area means different habitats. So the origin is different, their origin different, but they perform same functions, and it leads to convergent evolution. Convergent evolutions. So here in questions only, here giving an example of structural similarities between placental mammals. Structural similarities are seen. Structurally they are similar. Okay, so but their origin is different. Structural similarities between placental mammals and Australian marsupials. Okay, so here they perform same functions in geographical different isolated geographical different habitats. They perform same functions, but their origin is completely different. Such functions which leads to evolution, that is convergent evolutions. So here right answer is convergent evolutions. Okay, next one here, 161, the covering of ovum at ovulation. Okay, these questions which is framed from in human being. So generally in female ovulations, which is framed from ovulation phase. So generally ovulation phase is a phase in menstrual cycle. Okay, So we know that the in menstrual cycle there are four important phases are there. We will discuss later. So in that, third step is ovulation phase, ovulatory phase or ovulation phase. So this generally ovulation phase which occur at the 14th day of menstrual cycle. Okay. So ovulation phase which occur due to LH hormone peak or maximum. So when the luteinizing hormone which is maximum, which favor this maximum release of LH hormone from anterior pituitary gland, which favor the ovulation process. So once ovulation will occur, ovulation means here release of ovum or also called eggs from ovary, the process called ovulations. Okay. So release of ovum or egg from ovary, the process called ovulation phase. Ovulation phase occur at exactly 14th day of menstrual cycle, and this ovulation phase occur due to high or maximum amount of LH will release. And then this ovulation phase will occur in which ovum is released now. So this ovum or egg at ovulation which represent secondary oocyte. The released egg which represent secondary oocytes. In secondary oocyte condition only these eggs will be released. So once the egg is released like this secondary oocyte, and this egg form a new membrane. From this egg, it form the new membrane around it. So here the secondary oocytes are egg or ovum, which is form a new membrane from the membrane of ovum only. So new membrane will form from the membrane of ovum. So this new membrane is called, and this new membrane which is formed only inside of the inner side of the secondary oocyte or egg or ovary. So this new membrane is called zona pellucida. And outside of the cells, and next there are some various outside of the cells are present, and these cells are called corona radiata. So corona radiata present outside of the egg, and zona pellucida is present inner side of the egg. So this zona pellucida which is formed from the membrane of egg only. Okay, zona pellucida and corona radiata. So this is outside of the egg, and this is inner of the eggs. Okay, so like that there are few membranes which is formed around the egg. So here asking the covering of ovum at ovulation. So at ovulation stage, so which cover the egg means zona pellucida. So here zona pellucida is the right one. Why? Because zona pellucida which form after ovulation, at this ovulation stage, when the ovum which comes out from the ovary, and then it enter into the fallopian tube, in that stage only this ovum form a new membrane around it. That new membrane is only nothing but zona pellucida. So right answer is zona pellucida. Okay, next 162. Which of the following statement are correct statement we have to identify? Here three options are there. Energy flow from producers to the consumer is unidirectional. Energy pyramid can never be inverted. Transfer of energy follows 1% law. So we have to choose the correct answer from given below here. And these questions which is from ecosystem or ecology topic. So in ecology they focus on energy flow in ecological pyramid. Let's start some important notes on these questions. Okay, here generally the energy flow, here I'm writing energy flow through trophical level. Here we have various trophical level like this, like trophical level 1, 2, 3, 4, like that. We have various trophical level. So here the flow of energy through various trophical levels are strictly unidirectional. Very important. Unidirectional, not bidirectional. Only unidirectional. Means first, for example, in tropical level 1 we have producers. Means they produce food like plants. And in tropical level we have consumers. They consumes. In consumer again we have different types of consumers like herbivores, carnivores, primary carnivores, secondary carnivores, like that we have. So here the from the energy flow from producers to consumers only, means T1 to T2, and again the energy will goes from T1 T2 to T3 to T4, like that. The energy flow only in one directions, that is called unidirectional. Means here the energy which flow from producers to consumers only, not consumers back to the producers, only flow unidirectionally, producers to consumers. Okay. And coming to one another statement here, this energy flow from producer to consumers is unidirectional. Yes, this statement is true statement. Energy pyramid can never be inverted. Yes. So pyramid of energy always upright. See, pyramid of energy flow is only upright. If it is inverted, means then it be bidirectional, but energy flow is always and strictly unidirectional. Hence the pyramid of energy always upright only. Pyramid of energy means energy flow always upright only, always upright. It never be inverted. Pyramid of energy always upright, it never be inverted. Okay, means energy pyramid can never be inverted. So this statement also true. So here, when the flow of energy which occur in each successive trophical level from T1 to T2 to T3 like that. So remember, transfer of energy, here I am writing transfer of energy follows Lindman's 10% law of energy. 10% law. Okay, it follows the Lindman 10% law of energy flow only. But because here, from the transfer of energy from trophic level 1 to trophic level 2, only 10% of energy will transfer and 90% of energy will be lost in the form of heat. For example, from producers to consumer, primary consumer, only 10% of energy will transfer and the rest of the energy will last. And again, from primary consumers to secondary consumers, only 10% of energy will transfer. The remaining 90% of energy will get lost in the form of heat. So like that, so transfer of energy follows only 10% of law. So this law 10% of energy flow was proposed by Lindman, hence also known as Lindman's 10% law of flow of energy. So it follows 10% law only. So this was proposed by the Lindman, hence this law also called Lindman's 10% law of energy. Okay, so here transfer of energy follow 1%. So this statement is wrong. So here choose the correct answer, A and B. So in A and B, where we have A and B. So fourth answer is the right answer. Next one, 163. How many thekas are present in each lobe? Here asking each lobe of a typical bilobed angiosperms anther. So here this questions which is framed from sexual reproduction in flowering plant. In that they are focused on anther anatomy. Anther. So anther are also called microsporangium. So anther or microsporangium is a part of male reproductive organ. So that is stamen. So generally stamen has like this stamen. It is a stamen, male reproductive sex organ, in which here we have anther or microsporangium, and this one is called filaments. So filaments and anthers together form a stamen. So group of stamens together form male reproductive organ, that is androecium. Okay, fine. So here we need to discuss anther. Generally, anther in angiosperms are bilobe, means it has two lobes. For example, like this. So 1 and 2. So bilobed anthers are present generally in angiosperm. And in Malvaceae family only we have, in Malvaceae, here note, in Malvaceae we have only unilobe. Okay, unilobed anther are present. Uni means single lobe. Only we have single lobe. Okay, but in angiosperm, that in most of the angiosperm plants has bilobed anther like this. And each lobe here, each lobe, if we cross this lobe and we need to cut these lobes, then here we have find like this structures. So we will cross like this. So here each lobe, this one right side lobe and this one left side lobe, and each lobe contain two thecas. See, each lobe contain two thecas. So upper and lower, here also upper and lower. So finally, in bilobed anther contain four theca. Bilobed anther contain how many theca? Five lobe theca. Four thecas. Okay, tetra. But here in questions they are asking about each lobe consists of how many thecas. So each lobe means this one, this part. So each lobe contain only two theca. Hence this is called dithecous anthers are present in angiosperm. Okay, so each lobe means two theca. Entire anther means four theca. So if you read these questions, in questions asking about entire anther or each lobes. So here question asking about only each lobe of the bilobed angiosperm. So each lobes contain only two theca. If in questions asking about entire anther contain how many thecas, means four thecas. So right answer here two only. Okay, next one, 164. Which of the following statement is correct about plasmodium? So about plasmodium, these questions which is from the human health and disease. In that human health and disease, they specify one important protozoa organism, that is plasmodium. Remember, plasmodium cause malaria disease. So in plasmodium, here asking life cycle. So life cycle of Plasmodium, they frame these questions. Yes. So here first we need to read gametophytes. Here gametophytes develop in mosquito gut. Fertilization take place in mosquito gut. So development of gametophytes in mosquito gut and fertilization also taking in mosquito gut. Reproduction sexually in liver cell. Reproduction sexually in RBC. So then we know that Plasmodium perform its life cycle in two hosts. Perform its life cycle in two hosts. In human, it perform its life cycle in human first, and then it perform its life cycle in mosquito. In mosquitoes, the primary host is human, and the secondary host is the mosquito. So like that, Plasmodium perform two life cycle. It perform its life cycle in two host organism. One is human, and second one is in mosquito. So let's start in human. What happened? So what exactly will happen in human? So in life cycle of Plasmodium, generally here gametocytes are sexual stages. Gametocytes are nothing but their sexual stages develop here. I'm writing in life cycle of Plasmodium. This here gametocytes are sexual stages. These gametocytes are sexual stages which develop inside the human host. So in human host, where in RBC, only in RBC, red blood cell or erythrocytes only, these gametocytes at sexual stage can develop. And when female Anopheles mosquito which bite, so when female Anopheles mosquito which bite the infected person. So here gametocyte or sexual stages are developed in human host, means definitely this host become infected host. So here infected host. Okay, why? Because in which only RBC get, in RBC cell only this gametocyte development will be taking place. So here what happened, when a female Anopheles mosquito, remember, when female Anopheles mosquito which bites the infected persons, then what happened, these gametocytes present in RBC of human enter the mosquito body. So when the female Anopheles mosquito which bite the infected persons, then these gametocytes in RBC which enter into mosquito bodies, which enter into the mosquito body. So in mosquito body, what happen, when this gametocyte which enter into the mosquito body, so where only these gametocytes, male and female gamete, are undergoes fertilizations. See here, the fertilization are taking place in mosquito, but the development of gametes are taking place in human beings. So like that, in a human is a play important role for this gametocytes development of Plasmodium in RBC and liver also. But their fertilization which taking place in the mosquito body. So not only fertilization, and after fertilization the resultant structure is zygote now. So the zygote development also taking place in mosquito gut. Also taking place in mosquito guts. So remember, in mosquito gut what happened? Fertilization and development of gametes. Fertilization means formation of gametes and development of gametes in mosquito bodies. But in human RBC cells only what happened? Gametocytes development will happen. Okay, clear now? Right. So if you analyze these questions, so which one is the correct answer? So gametocytes develop in mosquito? No. So where gametocytes develop? In human body, that especially in RBC. Fertilization take place in mosquito gut? Yes. So here fertilization take place is mosquito gut. This one right statement. Still we need to discuss reproduction asexually in liver cell. No. So here generally parasites means Plasmodium. Here Plasmodium multiply inside liver. Okay, inside liver and asexually only. This Plasmodium which multiply in human body asexually only in liver and RBC, not sexually. So reproduction sexually here point out it. Okay, so this statement. So remember, in human being this Plasmodium parasites multiply only asexually. So this statement is wrong. Here this statement is also wrong. Not sexually. In human being always reproduction always a sexual method. But in mosquito only the reproduction is sexually. So remember that important note. Here in human the reproduction is a sexual mode only. But in mosquito the reproduction is sexually. So these two statement also wrong. So the right statement is second one. So 164 answer is second one. Okay, next one 165. Here read these questions. For a person with blood group A, which of the following is not, here keyword. So here blood group is O, and which of the following is not a possible combination of parents blood group. So not possible of parents blood group genotype. So here the person having O blood group means the progeny, but it is not a combination of parent. So we have to identify in which combination here the persons having O blood group. Okay, so we will discuss each and every concept in detail. So first, these questions which is framed from the principle of inheritance. Principles of inheritance. Principle of inheritance. In principle of inheritance, they focus on abio blood group. So let's discuss some introduction regarding to the abio blood group. So here, the abio blood group concept was given by a scientist called Carl Landsteiner. So Carl Landsteiner proposed this abio blood group. Okay. So generally, this abio blood group, which is present on ninth chromosome of I gene. So here ninth chromosome contain I gene. For example, think it is a ninth chromosome pair. So where we have seen I gene. So here we have seen I gene. So this I gene is responsible for this I gene is responsible for blood grouping. That is ABO blood grouping. This I gene is responsible for ABO blood group. This I gene present on the ninth chromosome. Remember. Note here, important note. Generally, according to Mendel, here, according to Mendel, he said every gene has only two alternatives. Every gene has only two alternatives. They are called alleles. For example, if one gene has only two alleles, but here exception of this Mendel. In this I gene has three alleles. It has three alleles. If a gene has more than two alleles, such alleles are called multiple alleles. Here, I gene has three alleles. As for according to Mendel, every gene must and should contain only two alleles. But here, this concept was exclude from this Mendel, or deviation from the Mendel. So in here, I gene contain three alleles. So they are like IA, IB, and I. So like that, there are three alleles are present, and this concept is called multiple allelism. So ABO blood group shows multiple allelism. Okay. So here, in exam point of view, here we need to identify a formula. We have different formulas for ABO blood group to identify their genotypes. So number of genotypes. So yeah, here I am writing. Just wait. Yes, here. So to identify number of genotype. So we have one formula, very important formula, that is n into n plus 1 by 2. Here remember, n is always represent alleles, number of alleles. N represent number of alleles. Here formula for genotype is n into n plus 1 by 2. Okay, and also we have formula for to number of phenotype. To identify the number of phenotype, physical appearance of character and genetical makeup of the character is called genotype. Physical appearance of the character is called phenotype. So physically, persons have four different blood groups. So for that, we also have one formula like n plus 1. So if we apply this n into n plus 1 by 2, so here I gene has how many alleles? 3 alleles. So here 3 plus 1 by 2. So here 3, 4 by 2. So 2 4 the 2 is 4. So 3 2 is 6. So here how many genotypes this ABO blood group have means 6. And coming to number of phenotypes, here n is 3 plus 1. So is equal to 4. So we have 4 what phenotypes. So finally here 6 are genotypes, 4 are phenotype contain this ABO blood group. Okay, and genotypes are like this. So how they are 6 genotypes are present means they have IA and IA. So these two genotype for A blood group, and also contain IBI. So these two genotype for B blood group, and here IA IB. So it is only one genotype for AB blood group, and coming to IEI is one genotype for O blood group. So like that, and A B all these are what? 4 phenotypes, and all these are like 1 here, 2 1 2 4 5 6. All these 6 are what? 6 genotypes they contain. Okay, fine, right. So now our primary duty is to identify. So the person is having O blood group, but their parent do not have this combination. So we have to identify. We will discuss how to identify all these things. Okay, so this is somewhat introduction regarding to the ABO blood group. Okay, fine, right. Sorry, yeah, we'll take another. We need to remove here. Yeah. So here first in options, giving first option okay, we will see first option here. Father has here, father first option here, option first, father has IB versus mother has IB, means father and mother both having B blood group. So we'll check out later, we'll do. And we need to take another option also here. So here in this, father has IA means B blood, sorry, AB blood group, and mother has IA means A blood group. Okay, first we'll do this two. So the person must and should here contain a person with blood group O must and should be present. Okay, here see. So if we make this, so when here IA IB, so here IB versus IBI. So here we have two heterozygous in condition, hence we have two gametes IB and I, and here also in heterozygous condition, hence IB I. So we have to do cross multiplication IB and also IB I, and here IB I, here I. So then we have to find out their phenotype. IB means B blood group, IBI means B blood group, IBI means B blood group. Here II means O blood group. Okay, so here II is O blood group, the person having O blood group. Okay, fine. So then we find out here second one. So here IA IB means definitely AB blood group, and both are heterozygous in condition, hence we have to do like they separate both gametes IA IB. Here they separate IA I. So here we need to do cross multiplication. Here IEA means A blood group. Here IAI means A blood group. Here IIB, here AB blood group, and IBI, here B blood group. So A, two A blood group, AB and B blood group. So here we don't have any, no O positive blood groups. Here O, and here, so here we have O. Here we don't have any, no O. So here no O blood group person will here. Not possible here asking. Okay, na, fine. Right. Next we'll take another combination. Here third combination is father has, wait, yeah, okay. Third combination here, father has IAI, mother has IBI. So yeah, we'll take here father has IAI, mother has IB, okay, and both are heterozygous in condition, hence the gametes are separate. I here from which there are two gametes are separate, IB I. Okay, so cross multiply. IA IB means B blood group. IB I means B blood group. Okay, so here IA IB na, so sorry, AB blood group. And here what we have, I na, so I sorry, first here we need to cross this one. I means B blood group, and finally I means O blood. Here we have O blood group. Okay, right, one more and last combination, we will check it. Yeah, so next here take this combination, father has IA and mother has I. So heterozygous in condition, both father and mother having A blood group. IA here also IA, so cross. IA means A blood group. IA also A blood group. IA A blood group, and II means O blood group. Here also we have O blood group. See, so in these questions there are three combination, the person having O blood group. At first combination O blood group is there, and third O blood group, and fourth combination O blood group. But in questions we have to find out which is the not possible O blood group person. So which is not possible to O blood group means the right answer is second. Why? Because if the father having IAIB means AB blood group, and mother having IAI means A blood group, father AB, mother A blood group, and these combination persons, they do not produce O blood group progeny. Remember? So remember here, AB father and A blood group mother. AB father, A blood group mother combination, they do not produce any O blood group progeny. So here not asking now. So then the right answer is second one. So 165 right answer is second one. Which of the following is used as a clot buster? So these questions from microbes in human welfare. So in this topic they mainly focus on microbes used as bioagents. Sorry. So microbes used as a bioactive agent, in which this questions which is framed. Okay. So in which bioactive agent used as a clot buster? Your question, clot buster. Right. So here let's start some explanation. So NCERT generally provided three highly important bioactive agents. Here, three important bioactive agents. So we'll discuss each type in detail. So in that, first one is strepto. So here the streptokinase, which is produced from a bacteria. Here the bacteria is strepto. So generally streptococcus are pathogenic, but this streptokinase, which is produced by modified this streptococcus, genetically modified organism it is. So here we need to modify their genes. So then we can obtain this enzyme streptokinase bioactive agent from this streptokinase. So remember, streptokinase is a genetically modified organism from which we obtain this streptokinase. And the importance of this streptokinase enzyme. So generally this streptokinase enzyme used as clot buster, very important. So used as clot buster. Here clot buster means to remove clots from the blood vessels. To remove clots from the blood vessels of patients. So who here from which patient means remove clots from blood vessels from the patient who are suffering with myoinfarctions. Very important, so the person who suffer with this myocardial infarction, or simply we can use heart attack person. Heart attack persons are myocardial infarction, so in which we have to remove the clot from their blood vessels. Okay, so likewise we use streptokinase enzyme as a clot buster, remember. Now coming to one more important bioactive agent, so that is cyclo A. So the cyclosporine A, which is produced from fungus. So here which fungus means very important fungus here, Trichoderma palli. So from which fungus we obtain this cyclosporine A. So this cyclosporine A used, which is used as immunosuppressant. Very important. Cyclosporine A used as immunosuppressant in organ transplanted patient. The patient who undergoes organ transplantation. So in that person we can use the cyclosporine A to suppress the immune system. Why? Because when we donate, when there is a organ transplantation from one organism to another organism, so the patient who admit the organ, so his own immune system it cannot recognize this organ. So then what we need to use, we use the cyclosporine. So the cyclosporine which suppress the immune system of the patient. Okay, right. Now third one, here I am writing the third one, that is statins. So statins is also very important bioactive agents which is produced from the microbes, and which microbes it is produced means very important, fungus. It is also fungus. In fungus especially which is produced from yeast. So do you know yeast is a unicellular fungus. Generally fungi are multicellular, but yeast is the only fungi in unicellular in nature. And from this yeast we can obtain the statin. From which yeast? In yeast we have different species in Monoscus purpureus. So from this yeast we obtain statin. So the statin used as, it is used as which will lower the blood cholesterol level. Very important. Lower the blood cholesterol level. Which lower the blood cholesterol level. So likewise there are three important bioactive agents are produced from various microbes like streptokinase, cyclosporine, statin. So read this option. So which is used as a clot buster here? Which one? Streptokinase. So right answer is streptokinase. And statin used as which lower the blood cholesterol level. Cyclosporine used as the immunosuppressant. So here not sorry. Which of the following is as a health? There are various common STD diseases are syphilis, POTS disease. The right one. And not, sorry, here ask in question ask not sexual transmitted disease. See, gonorrhea is a sexual transmitted disease, genital warts is also sexual transmitted disease, syphilis also sexual transmitted disease. So which is not sexually transmitted disease means this one, tuberculosis. So right answer here is tuberculosis. Now we discuss what exactly mean of how tuberculosis occur, and this tuberculosis which caused by the bacteria. Okay, it is bacterial disease, but it is not a sexual transmitted disease. And how they are transmitted means air, through air, in air droplets, through the droplets only this disease will spread from one person to another person, but it is not a sexual transmitted disease, bacteria, tuberculosis. So tuberculosis generally we call TB. So this caused by a bacteria called Mycobacterium tuberculosis. Mycobac tuberculosis. Mycobacterium tuberculosis. Okay, so your bacteria name is Mycobacterium tuberculosis, which cause TB or tuberculosis. It is a bacterial infection, and this bacterial infection which cause only air or droplets. It is a airborne disease. Droplets by sneezing and coughing. So when we get cough and sneezing, so during which we have released some droplets. So these droplets through air is transmitted to the healthy person from infected person. So likewise, tuberculosis is a airborne disease, not a sexual transmitted disease. Okay, right. So remember, TB day observed on 24th March. Remember, very important. On 24th March we observe TB day or tuberculosis day. Okay, next one here, 161 statement type of question, assertion and reason type of questions. So once read this assertion: in experiment, Mendel observed that F1 progeny or F1 generation plant all tall, none are dwarf. Stem height is contrasting trait, with tall being dominant and dwarf being recessive. So we have to choose most appropriate answer from these options. So these questions which frame from principle of inheritance. Principles of inheritance. So which mainly focus on Mendel hybridization experiment. Do you know about Mendel? So Mendel was considered as a father of genetics. Mendel considered as a father of genetics. Mendel did an experiment called hybridization experiment. So Mendel experiment is hybridization experiment. So he worked on pea plant. The pea plant means scientific name Pisum sativum. So he worked on hybridization experiment on pea plant Pisum sativum, and then he observed, very important, 7 characters. He observed 7 character and 14 contrasting trait. 14 contrasting traits he observed on pea plant while he performed hybridization experiment. And also he proposed, by doing hybridization experiment of pea plant, he proposed a law or theory. So the law is law of inheritance. He proposed law of inheritance. Okay, and he proposed 3 law of inheritance. He proposed 3 law of inheritance. And that first one is law of dominance, and second one is law of segregation, or also we can use law of purity. And also he proposed law of independent assortments. So likewise, there are 3 laws are proposed. So remember the first 2 laws, this law of dominance and law of segregation, which explain monocross or one gene inheritance. Monohybrid cross also called one gene inheritance. And the law of independent assortment, which explain dicross. So means 2 gene inheritance, also called 2 gene inheritance. Okay, so this is all about the some importance of the Mendels. So now we move on these questions here. Here, generally in this experiment, Mendel did experiment monohybrid cross. He performed monohybrid cross experiment. Monohybrid cross. So here monohybrid means crossing of taking up one character only. So here, which character here taking? Stem height character here too. So here I will explain in just bit. So here Mendel choose monohybrid class in which he choose stem height characters. So stem height character regulated by a gene called T, gene T. So in which stem height has 2 contrasting traits. Every character has its alternative forms. They are called traits. Remember, here I am, according to Mendel, here note. Here note is, remember 2 important terms here I will explain. One is first, every gene, according to Mendel only. According to Mendel, what he said, every gene has 2 all forms. They are said to be as a alleles. And remember one more important note, every character also has 2 alternative forms, and they are said to be as a traits. So here there is a clear cut differences between allele is different, trait is different, gene is different, character is different. Every gene has 2 alternative forms. They are said to be as a alleles. Every character has 2 alternative forms. They are said to be as a traits. Remember one more important note here. So every gene is responsible for character. So here one gene responsible for one character according to Mendel. Okay, fine. Every gene responsible for one character. For example, here the gene is T, which responsible for which character? Stem height. So here T is the gene, stem height is the character. It is a gene, it is a character. And just now we said every gene has 2 alternative forms like this. One is capital T and another one is small t. So every gene has 2 alternative forms, and these capital T and small t, capital T, small t's are traits. Traits, alleles. Every gene has two alternative forms, they are called alleles. Likewise, every character has two alternative forms. Like stem height has two alternative traits. Here stem height is a character and it also has two alternative forms, they are said to be as a traits. So likewise, stem height has tall or dwarf. Tall or dwarf. Here tall or dwarf are what? Tall or dwarf are traits and stem height is what? Characters. So likewise, every character has two alternative forms, they are said to be traits. Here T is what? Gene, and capital T small t is what? Alleles. So every gene has two alternative forms. Okay, likewise, remember. So like stem height is a character, it regulated by T gene. Here stem height has two traits. Yes, they are tall and dwarf. So every here tall was expressed by capital T allele and dwarf is expressed by small t allele. And these two are homozygous in condition, means same kind of chromosomes are present. And if they are in homozygous, they produce only one gametes and one chromosome, like capital T and here from small t. If they are heterozygous, like capital T small t, then it produce two different gametes, capital T and small t. But here tall has capital T, dwarf has small t, means they are in homozygous in conditions. And here these two gametes are undergoes fertilization to form capital T and small t. So here the capital T, here capital T which dominate over the small t. So here we get one first progeny or F1 generation. So if you observe the F1 generation product, here the progeny or the offspring always tall. So this statement was said by the Mendel only. So F1 progeny must and should be only dominant. So here tall is nothing but dominant. Why? Because capital T dominate over the small t, tall. Okay. So by analyzing all these things, we can once read this accession and reason type. So in accession, what here giving experiment, Mendel observed that F1 progeny plants all are tall, none are dwarf. Yes, in F1 progeny we get only tall plants. So this accession A is right statement, correct statement. Now coming to reason 2, reason stem height is contrasting trait. Yes, with tall here being dominant. Here tall is being dominant, dwarf is being recessive. Yes, here tall is being dominant and dwarf is recessive. Means and both statements are right. Assertion and reason both are right. So here we have to choose both A and R are correct, and R is the correct explanation for the A. So right answer here, third one. Okay, right. Yeah. Next one, 169. The method of direct of injecting sperm into ovum in assisted reproductive technology. So here there are four options are there: ICSI, ET, GIFT, and GIFT. So here these questions which is from the reproductive health, from the health topic. So in reproductive health, which is mainly focused in infertility and ART, infertility and ART. So here ART means assisted reproductive technology, assisted reproductive technology. Okay. So what is infertility? Nothing but inability to conceive or inability to give birth is called infertility. And there are many reasons are there for infertility. Already we discussed all these things in a video lecture. So in my YouTube channel, once you go through and search infertility and ART by Praveen Naik sir, then there are various reasons are there. Already we made a video, please watch that. And here and there is a solution for infertility. Okay, there is a solution, means the infertility can be cured by using a technology. So that technology is only nothing but ART. Okay, so here ART means assisted reproductive technology. So here in ART, so we have different types of ARTs, like here we will discuss. So in which based on the fertilization taking place, if fertilization occur within the female body or outside of the female body. Here if fertilization occur outside of the female body, they are said to be as a IVF, in vitro fertilization and embryo transfer. Okay, and if fertilization will take place within the female body, then it's said to be as a here in vivo fertilization. Okay, so in vitro fertilization and in vivo fertilization. Remember, in vitro fertilization and embryo transfer, where fertilization occur, fertilization outside of the female body. So fertilization outside of the female body, and here fertilization inside of the female body. So that is a major differences inside of the female body. So once here see, in IVF and ET, again we can classify them into two categories. Here I am writing there are two types of in vitro fertilization or embryo transfer. In vitro fertilization also we can use another term, test tube babies. Yeah. So there are two types of in vitro fertilization, like there GIFT and one more is IE. So these two are types of IVF and ET. So here GIFT means zygote intrafallopian transfer. Here zygote intra transfer. Okay. Here IUT means, here I am writing, here IUT means intrauterine transfer. So what is the major differences between GIFT and intrauterine transfer? Means simple. So in which here zygote are early embryo. So the fertilization taking place outside, means in laboratory only or in test tube only fertilization taking place, and after fertilization the resultant structure is zygote. Or zygote later develop into embryo. Or zygote are early stage of embryo, means the embryo less than 8 blastomer stage. The embryo less than 8 blastomer stage of embryo. Here what we need to do, we need to transfer this zygote or early embryo less than 8 blastomer stage directly into the fallopian tube. Here directly transfer into fallopian tube. Here we can transfer zygote or early embryo less than 8 blastomer stage directly into fallopian tube. In name only it is giving suggesting. Here see, zygote means zygote or early embryo. So where we transfer in fallopian tube, but the fertilization will occur in outside of the female body. Okay, only we have to do after formation of zygote in outside, then we have to collect those zygote or early embryo, then we need to transfer in female body only in fallopian tube. So that method is called GIFT. Now coming to IUT, so in which name only suggests intrauterine transfer. In uterus only we can transfer, but here the most thing is in which embryo here will transfer, and embryo must be more than 8 blastomer stage, like 8 or 16 or 32 blastomer stage embryo only transfer in IUT. So less than, more than, here more than 8 blastomer, like 16 or 32 blastomer stage of embryo only transfer in uterus. Here where we transfer in uterus. So here we transfer embryo that more than 8 blastomer stage directly to the uterus. So that method is called IUT. So likewise we have different IUT method. Now coming to in vivo fertilization, also we have different methods like GIFT method. Okay, and here ICSI method, and also we have AI method, and also we have IU method. Likewise we have different methods. So in simply here we'll discuss here in GIFT method, generally gamete intrafallopian transfer. So here only gamete only transfer to the female body, that is female fallopian tube. In fallopian tube only we can transfer the gamete. So whose gamete? Generally sperm gamete, sperm or ova gamete. So male gamete, here male gamete or female gamete only we can directly collect, and then we can directly transfer to the fallopian tube. So in why fallopian tube we transfer? Why? Because in fallopian tube only the fertilization taking place. Okay, so like that. And coming to ICSI, is it, here fertilization is taking place inside the female body. Just what we are doing, just we have to collect gametes either from sperm, either from male and either from female. Okay, that's it. And then we directly injected that gamete after collecting into direct fallopian tube. In fallopian tube only the fertilization process will be occur. So ICSI means intracytoplasmic sperm injection. Here ICSI means it is the most common method which is applied, which is performed to the male only. Remember, it is male only when the male having fertility rate so high, severe, so severe male fertility factor. So when the male person have severe male fertility factor, those persons can only undergoes this ICSI method. Very important. ICSI method for whom the person having severe male fertility factors. Male fertility means in which there are number of fertility factors will include, like sperm count will be azoospermia, zoospermia, like that. There are severe male fertility factors are influenced to go for ICSI method. Okay, so in which what happen, in which what they will do means, in which sperm is directly collected from testis. Sperm is directly collected from the testis and injected directly into the egg, okay, or ovum. So collection of sperm directly from the testis of male, and after collect, then directly we injected that sperm directly into the egg or ovum. So that process is called ICSI. So if you observe all this method, then here read. So in which what here asking, so which method of direct injecting sperm into ovum in assisted reproductive technology means ICSI. Here just now we discussed. So ICSI is the right answer. And embryo transfer is a fertilization occur outside of the female body, so it is not related. It is IVF. Okay. And gamete intrafallopian tube. So gamete only we have to collect gamete either from male and female, and it is also in vivo fertilization. It is a in vivo fertilization, but embryo transfer is a in vitro fertilization. Here zygote intrafallopian transfer. Here GIFT is a in vitro fertilization. So here, so we won't use second, third, and fourth. Only we use ICSI method to collect directly sperm from the male and directly injected into the egg or ovum. So right answer is 169. Right answer is first one. So next one, sorry. So 170. The inactive form of Bt toxin. Here inactive form of Bt toxin is converted to the active form of toxin in insect gut by protease enzyme, by nuclease enzyme, due to alkaline pH, due to acidic pH. Okay. And here this questions which is from biotechnology application topic. Biotechnology application. In biotechnology application, which is mainly focus on Bt toxin, Bt crop. So here we will discuss in detail of Bt. Here about Bt. Here, so Bt toxin protein existed. Bt toxin protein exists as an inactive protein. Generally, it is an inactive protein. Inactive pro. So within a bacteria, in which bacteria which is exist as a inactive protein means the bacteria is called Bacillus thuringiensis. So generally, this Bt toxin produced from a soil bacteria called Bacillus thuringiensis. It is a soil aerobic bacteria. Soil bacteria. Okay, soil bacteria. So in soil bacteria Bacillus thuringiensis, the Bt toxin protein exists as a inactive protoxins. How they are exist as a inactive pro? Here, once this insects which ingest this inactive protoxin, then this inactive protoxin get converted into active toxins. Remember, so inactive protoxin get converted into active toxins when the insect ingest this inactive toxins. So these active toxins, what will do? This active toxins binds, and this conversion of inactive toxin converted into active toxins. And due to here, why how this inactive form of protoxins converted into active toxin means very important, due to alkaline pH of here, alkaline not acidic, alkaline pH of insect gut. Due to alkaline pH of insect gut, here the inactive form of protoxins converted into active toxins, active Bt toxins. And once they are converted into active toxins, and these active toxins binds to the midgut epithelial cell. Remember, which is bind to the midgut epithelial cell. When this active Bt toxin which bind with midgut epithelial cells, then which create pores, means holes, and these holes which cause the insect to death. Very important. So likewise, the insect get killed by these Bt toxins. Okay. So before, in Bacillus thuringiensis, this in Bacillus thuringiensis, a toxin which is present in inactive form like inactive protoxins, and when this inactive protoxin form of toxins which ingested by the insect, and then in insect, due to presence of alkaline pH of insect gut, then inactive form of protoxin is converted into active form of protoxins. Sorry, not active form of Bt toxin, and this active form of Bt toxins which binds to the epithelial cells of midgut, and due to which create a pore. This toxin create a pore in epithelial midgut, and due to creating pore, then the insect get death. So this is how this process will occur. And here the right answer is, so inactive form of Bt toxin is get converted into active form of toxin because of alkaline pH. So right answer is third one. Okay. So here we need to remember some important points. So there are generally these Bt toxins works on some specific group of insects only. Okay. So and there are highly, the gene which is responsible for producing Bt toxins also is differ. So we will discuss about Bt toxins are effective against, generally here Bt toxins generally get effective. Bt toxin get effective against specific group of insects, not all group of insects, only which is effective on specific group of insects. So what are they? So here we will discuss. So there are specific group of insects on which only Bt toxins get work effective. So here I am writing the first group and most important group on which the Bt toxins work effectively. So they are lepidopterans, and these lepidopterans which include two insects like tobacco budworm and also armyworm. So these two insects belongs to this lepido group of insects. Now also this Bt toxins works on another group of insects like coleopterans. So in coleopterans include beetles, and one more specific plant group, insect group, that is dipterans. So here dipterans are include flies and mosquitoes. So especially on this specific group of insects only get effective on this against Bt toxins. So these are the various insects. Okay. So one more important point here I want to explain. So one more important points we need to remember on Bt crop here. Specific Bt genes. Here we need to discuss specific Bt genes. So just now we discussed specific group of insects on which the Bt toxin work effectively. Now here we will discuss specific Bt genes and their crop. So the questions which may be framed from this specific Bt genes and crops. Okay. So remember one important note here. I am writing here note. So Bt toxins coded by, so these Bt toxins generally coded by or expressed by a gene called cry gene. So very important. This cry gene play important role to code the Bt toxins. Okay, cry genes. Yeah. So here we go. So here specific Bt genes like cry and cry. So these specific genes which works on special like some crops like cotton boll against cotton bollworm, and one more cry. Remember cry1 AB genes, and this cry1 AB genes which control the corn borer insects. So cry1AC, cry2 here, cry1AC, cry2AB. Don't confuse, cry2AB is cotton ball, cry1 corn borers. Okay, so these genes control cotton bollworm and cry1AB control corn borer. Okay, so this is all about the 170. Okay, next one 171. Given below are two statements. Statement type of questions like assertion and reason type. Here assertion reason. Assertion type of question here. Logistic growth model of population. Here asking about logistic growth model of population is considered more realistic than the exponential growth model. And the reason is resources are finite. Finite means limited. Limited resource. Okay. Once we analyze this statement. So here these questions which is framed from organisms and population. So in this they mainly focus on growth model. In organism and population we have two growth model. They are like logistic growth model of population, and also we have exponential growth model of population. So here in both which is given. So here the logistic growth model in which the main important concept here resources are limited or finite. Here in exponential growth, the resources are unresource and infinite. Okay, remember the major differences between logistic growth and exponential growth. In logistic growth is resources are limited, finite, and in exponential growth the resources are unlimited and infinite. Okay, right. So here, so when there is a what exponential growth of model here I'm writing about exponential growth model. So here we know that in exponential growth model which assume what, so resources are unlimited resource, so which rarely happen. So generally in nature we don't have unlimited resource. Sometime only rarely this exponential growth will occur. Okay, why? Because resources are limited only. Okay, and here and in real world the resources are limited only. Okay, and due to limited resource in nature, important word here, in the nature resources are limited, but very rarely there are unlimited resource. Very rarely happen unlimited resource, but mostly 99% of the resources are limited resource only. Okay, so due to limited resource there is a competition between individuals. Yes, when there is a limited resource there must be a competition between in of the same species. So what happened when there is a competition? So when there is a competition, definitely only one, those who have fittest for this survival nature, the only which we have fittest of the survival, they can only survive, and another species get loss or die, or they can exclude. Okay, so here this competition between individual is mainly due to these resources are limited resources. Okay, so in which the species which have the like have the fittest fit to the survival, they can only survive and they can only reproduce, and another species get completely exclude, or they can completely migrated, or they can completely extinct. Okay, they are survive and they are reproduce, and they only these fittest of the survival organisms only carrying maximum carrying capacity, they only carry maximum carrying capacity indicate with K. Okay, so another species get completely extinct. So here the nature only play important role, so which species to be survive and which species to be extinct. So the species, the individual organism which have fit, then fittest to survival, then only they survive. Why? Because the all this whole process is due to resources become limited resource. If there is unlimited means the number of species get, there is no competition between individual, so they can easily survive. But here due to limited resource, thus in between species there are, and between individuals there are competitions, in which competition the strongest animals, the strongest individuals only have fit to the survive, and other one is can extinct. Okay, so this statement, so therefore here conclusion is the logistic growth is model, logistic growth model of population is much more realistic than exponential. Why? Because generally this exponential growth is generally which not occur, which is rarely occur. So hence therefore here therefore remember, logistic growth model of population is much more realistic. It's much more realistic. Remember, it's much more realistic. Very important. So like that we have say the logistic growth model of population is considered more realistic than the exponential. Yes, just now we discussed why. Because in logistic growth model we have limited resource, so there is a competition between individuals, so in which the strongest and fittest animals only can survive, other one will become extinct. And in nature, in real world, the nature in the real world the resources are only limited. Remember. Okay, right, so this statement is right. And coming to resource are reason are resources are finite. Yes, so due to resources are finite, then the logistic growth is growth model of population is more realistic than the exponential growth. So right answer, both are correct here. Both A and R correct. R is the correct explanation for A. So right answer is third one. Okay, so 172, which of the following are secondary lymphoid organ? So these questions which is from human health and disease, so in which they mainly focus on lymphatic systems, lymph system. So lymphatic system is a specialized systems which is present along with circulatory systems, and in which lymphatic system the lymph which transport or carry. So generally this in lymphatic system we have lymphatic organs. So generally these lymphatic organs are classified into two categories. Okay, so lymphatic organs are classified into two categories. In that first one is primary lymphoid organ, and the second one is secondary lymphoid organ. Primary and secondary lymphoid organ. So we will discuss what exactly the differences between primary and secondary lymphoid organ. So in primary lymphoid organ what happened? So where in primary lymphoid organ, immature, remember, immature lymphocytes is a type of white blood cells. Immature lymphocytes get differentiated here, are differentiated, or also can we use mature. So here in primary lymphoid organ, immature lymphocytes undergoes differentiation or mature into how antigen cells. So here only in primary lymphoid organ only the immature lymphocytes differentiate into antigen specific cells. And generally this primary lymphoid organs are include like bone marrow, very important, bone marrow, and also thymus. So thymus and bone marrow both are comes under primary lymphoid organ. So in these two organ only the immature lymphocytes differentiate into antigen specific cells. Now coming to secondary lymphoid organ, so here what happened in primary lymphoid organ, immature lymphocytes get activated like differentiate into antigen sensitive cells. But here in secondary lymphoid organ, where the antigen sensitive cells means mature lymphocytes, here this mature, here I am writing, this mature lymphocyte cells get interact with antigen here interact with antigen, here nothing but pathogen, where antigen nothing but pathogen. In which region means secondary lymphoid. The maturation of complete maturation of lymphocytes taking place in primary lymphoid organ, but after mature the mature lymphocyte then move to the secondary lymphoid organ. In secondary lymphoid organ only they interact with the pathogen or antigens. Okay, and these secondary lymphoid organ include, here I am writing, secondary lymphoid organs include, like they include spleen, very important here, spleen belongs to secondary lymphoid organ, and lymph node, lymph nodes, and the third one is tonsils, and the next one is pairs patches. So these are the four important secondary lymphoid organ. So where mature lymphocytes interact with antigens, interact means they fight. So here this organ, this secondary lymphocytes act as an antibody. Generally it produces antibodies, lymphocytes produce antibodies, and that antibody fight with antigen only this region, secondary lymphoid organ. So this is all about the 170. So here we have to choose which of these following are secondary lymphoid organ. Here bone marrow is a primary lymphoid organ. Tonsils, so here tonsils is a secondary lymphoid organ. Yes, secondary lymphoid organ. Spleen also secondary lymphoid organ, but thymus is a primary lymphoid organ. Primary lymphoid, here see bone marrow and thymus both are primary lymphoid. So we have to choose only secondary lymphoid organ, that is B and C. Here in options where we have B and C, here at fourth one B and C is the right answer. Okay, B and C is the right answer for this 170 questions. So right answer is fourth one.